Welcome back. And in this second part, we're going to be talking about some of the molecules that are relevant in biology. So overall, there's two types of compounds. There's inorganic compounds. Inorganic compounds generally lack carbon. They're usually small and structurally stable. The other type is organic compounds. We're mostly going to be talking about organic compounds today. There's 4 different types that we're going to go over, and these always contain carbon. They have carbon as their skeleton, and they're structurally complex. And we'll see that here in a second. So here's an example down here of an organic compound. And then we also have these functional groups. So in addition to the carbon skeleton, so we see all this carbon here, carbon. We also have a functional group. A functional group is responsible for the properties of that organic compound. So here is a phosphate. Phosphate right here is part of this phosphate head. And we're going to see when we talk about phospholipids, which this is, that this causes this part of the phospholipid to be polar, which is what gives it its function. That's why it's called a functional group. This nonpolar tail is nonpolar, right? The chemical compounds of these molecules is determining their function. Before we get into organic compounds, we're going to very quickly go over acids, bases, and salts. So an acid is something that dissociates into a hydrogen ion. We have hydrogen ions released from an acid, and also a negative ion. A base dissociates in water into a hydroxide ion and a positive ion. And a salt is going to dissociate into a cation, which is a positively charged ion, and an anion, which is a negatively charged ion. And neither of those are going to be hydrogen or hydroxide. So acids and bases are measured through pH, and pH is the measure of the hydrogen ion concentration. So it's concentration of those hydrogen ions. As we mentioned, in an acid, it's going to release a lot of hydrogen ions. So this is going to be down here. We see we have a lot of hydrogen ions, and that's going to be the opposite with hydroxide ions. If we have a lot of hydrogen ions, we don't have very many hydroxide ions. And bases is the opposite. Remember, we said in a base, it's going to release a lot of these hydroxide ions. So we see a lot of those in a base and not very many hydrogen ions in a base. We also use this pH scale, or these numbers from 0 to 14 to measure the acidity or the basicness of a compound. So something that's very acidic is going to be a 0. Something that's very basic is a 14. And also something that's important to note here. Ooh, what happened? Oh, goodness. I don't know why it's being sketchy. I'll just tell you, though, something that's important to note is that it's logarithmic. So something that is acidity of 1 versus something that is acidity of 2 is not double. Or 1 is not double the acidity of a 2. It's 10 times the acidity of a 2. Something that has a pH of 1 is 100 times as acidic as a pH of 3, 1,000 times acidic as a pH of 4. So it's 10 times acidic each time you go down a pH number. Alrighty, so now we're going to get into the organic molecules. There's 4 of them: carbohydrates, lipids, proteins, and nucleic acids. Let's go through each of those. So starting with carbohydrates. So these can be part of the cell structure. So we will see carbohydrates in the cell membrane. And it's also a significant part of the energy source of the cell. Types of carbohydrates include sugars and starches, and they consist of the formula CH2O or some form of that. So for example, C6H12O6 is the formula for glucose. So it's always going to be some form of that formula. So the monomer for a carbohydrate is a monosaccharide. Some examples of monosaccharides include glucose, fructose, galactose, etc. These are always going to be in these like a cyclical shape here, or a ring shape. So all of these angles here are carbons. Okay, so there's a carbon ring here. If we put two of these monosaccharides together, we get a disaccharide. It's going to be linked here with the oxygen like we talked about earlier, the dehydration synthesis. Glucose and fructose makes a sucrose. Glucose, two glucoses makes a maltose. Galactose with a glucose is a lactose. And then we can link many of these monosaccharides together, and that will give us a polysaccharide. That can be a very long chain. Glycogen is an example of a long polysaccharide. All right, next is lipids. So lipids are the primary component of the cell membrane. Most lipids are non-polar, and anything that's non-polar is insoluble in water. So keep that in mind as we go forward. Anything that's non-polar, insoluble. Okay. Here is a triglyceride. So that's going to consist of a glycerol and 3 fatty acid chains, hence the name triglyceride. Another type of lipid is a phospholipid. And the phospholipid is what makes up most of the cell membrane. It's going to consist of a glycerol, so it's here in the middle, 2 fatty acids, and a phosphate group. So here's the fatty acids down here. This is what we call the tail of the phospholipid and the phosphate group. And as I hinted at earlier, the phosphate group is a polar region. So it has a charge. You see this negative charge here. Because of this negative charge, that's going to make it polar. Because it's polar, it's going to make it hydrophilic. These tails are non-polar, so they're going to be hydrophobic, and that's what makes them orient themselves in this way to form the cell membrane, or the phospholipid bilayer. We have the heads facing the outside and the inside of the cell, where they're going to be in contact with water, with the tails on the inside where they are hidden, right, from the water. Also, something else is saturated versus unsaturated fats. So a saturated fat, it has all single bonds between the carbons. And so something that has single bonds is going to be saturated with hydrogens. That's where that name comes from. In an unsaturated fat, it's may have some double or even some triple bonds. So here's an example of a double bond here. This isn't a chemistry class, so I'm not going to go into it too much, but when you do make a double bond, that makes where it's not going to be saturated with hydrogens. So that's where that name comes from. But what I do want you guys to really pull out of this is that when you start to add in double or triple bonds, that starts to break or bend this tail here. See where we have this bend here? The more bends that we have inside the tails, it's going to be more loosely packed. So here's an example of saturated fatty acids. So the phospholipids are very closely packed together. They're not going to move around as much. Something to keep in mind or kind of help you remember this is like butter. Butter at room temperature is typically a solid. Butter is also a saturated fat, and that's why it's a solid. It's because the saturated fats are more closely packed together, so it's a solid at room temperature. Whereas once we get unsaturated fats, we start to have more of these double and triple bonds. We start to have more bends in the tails. Then it's not going to be as tightly packed together. An example of this would be like oil. Oil is an example of an unsaturated fat. It's going to be a liquid at room temperature because it's not packed together as tightly. Another type of lipid is steroids. A steroid is going to have these 4 interlocking hydrocarbon rings. So it's going to have this shape. So if you see this shape, think about the steroid. All right. Mostly just want you to be able to recognize steroids. Also know that steroids, we have hormones that are steroids. We also have steroids in our cell membranes. So those are a couple of examples of steroids. Moving on to proteins. So proteins are very essential in structure and providing functions for the cell. So most of the things of the cell are going to be proteins. So if you ever have a question and you don't know, you're more than likely to get it right if you just guess protein. But some of the functions of proteins include enzymes. So enzymes are made of proteins. These are going to help to speed up those chemical reactions. They're protein catalysts. There's transporter proteins that are going to move chemicals across the plasma membrane. Flagella are made of proteins that aid in movement of the whole cell. And some bacterial toxins, not all, but a lot of them are made of proteins, as well as a lot of the cell structures. The monomer of a protein is amino acid. An amino acid is going to have 4 parts. We have the carbon skeleton here, just like all of our organic compounds have had. There's also an amine group, which has a nitrogen in it. There's the acid group over here, and the R group. Now, it's the R group that's going to vary. So we have all these different types of amino acid. Everything's going to be the same except for the R group. So glycine, aspartic acid, lysine. Note that the R group is the only part that's changing. We can put 2 of these amino acids together. They're going to be bonded by the peptide bond. That process is going to be the dehydration synthesis. And so the breakage of that is going to be the hydrolysis that we've talked about earlier. There's 4 different levels of structure to proteins. So the primary structure is literally just the order of those amino acids in the chain. So lysine, glycine, tryptophan, etc. Whatever that order is of amino acids, that's the primary structure. Secondary structure is as we can start to form bonds across these amino acids. So not directly from one amino acid to the next in the chain, but across the chain here. And there's 2 different secondary structures you need to know. So there's the alpha helix. It's going to be this like spiral structure here. There's also the beta sheets. So this is where this chain is just going to fold back on itself into sheets like that. Third is the tertiary structure. So this is the total globular structure of the protein. So we have one protein chain here, and the way that it all ends up folding up is going to be the tertiary structure. In our tertiary structure image here, we can see an example of an alpha helix here. See how it's that spiral? We also see some beta sheets where it's folding back and forth. We also see some areas of the chain where it's not in either of those two, but it's just folding back on itself. So the total chain, the way it's folded up, is the tertiary structure. And then if we're going to put more than one chain together to make a functional protein, then it's going to have a quaternary structure. Not all proteins are going to have a quaternary structure because sometimes some proteins only have one chain. But a lot of them have more than one chain, and that's going to provide our quaternary structure. So we're going to bring 2 or more polypeptide chains together. Protein structure is very important. If the structure or shape of a protein changes, it loses its function. And we call that denaturation. So if the shape changes of the shape of a protein changes, that's what we call what's denatured. Denaturation of proteins can happen due to changes in temperature or pH. Changes in temperature or pH can change these like hydrogen bonds. There's also sulfide bonds amongst those amino acids that's causing those alpha sheets, those beta sheets, any other of the other folds in the structure. Once those bonds break or change, because of changes in temperature or pH, that's going to cause the shape to change. Once the shape of the protein changes, it no longer has its function. Also enzymes. So here's enzymes. I said we would talk about them later. Enzyme is a protein catalyst. It's not going to be affected by the chemical reaction, but it's going to help to speed up these chemical reactions. So there's 3 steps to the enzyme function. The first step is the formation of the enzyme substrate complex. So this purple part here is the enzyme. These orange parts are the substrates. Those substrates are going to come in and bind to the active sites of the enzymes. Second step is the formation or breaking of these bonds. This is where the chemical reactions are actually occurring. So in this case, this example, they're showing the formation of this bond. And then the third step is the release of the product. The enzyme is unaffected, so now I can go and bind to new substrate and do the whole process all over again. So we talked about carbohydrates, lipids, proteins, and lastly, we have nucleic acids. The basic unit of nucleic acids is nucleotides, and it has 3 parts. We have a 5-carbon sugar. So this would be your deoxyribose or ribose here. We have a phosphate group. And lastly, we have a nitrogen-containing base. This base is going to be your A, T, C, or G. DNA specifically is going to have deoxyribose for the sugar. That's going to be in the middle. And then we also have the phosphate group. And then in the middle, this is like on the ladder part, or the where you step on in the ladder, that's going to be the base. And then it's a double helix. So we're going to have 2 different bases. So here's one base, and on the other side of the ladder is another base here. Okay. So keep in mind that we have a whole nucleotide here, a whole nucleotide on the other side, and then those nucleotides are linked on the side of the ladder. Now, if you just look at one side of the ladder, and you look at the order of those bases, T, G, T, etc., you go on, that order of bases is what provides the genetic instructions for the organism. Last thing we're going to talk about is adenosine triphosphate, or ATP. This is a very important and relevant nucleotide. Um, it's going to contain the same three parts of all nucleotides. We have the base. In this case, it's going to be adenosine. We have a sugar, it's ribose, and a phosphate group. But in adenosine triphosphate, we have 3 phosphate groups. This molecule is very important because it stores a large amount of potential energy. So when this bond here breaks, this phosphate bond, and it breaks and releases this last phosphate, that's going to release a huge amount of energy. And because it releases so much energy, it's because the ATP is the what we call like the money of the cell. It's the energy of the cell. Once we remove one of these phosphate groups, that's going to leave us with just 2 phosphates. So it's going to become ADP, or adenosine diphosphate. You're going to see this a lot as we move forward. So make sure that you understand ATP and how it stores this energy. A couple of examples of where ATP is used. So ATP is used to provide the energy for transport proteins. So ATP is going to provide, it's going to release one of its phosphates to provide the energy for this membrane protein that will end up having ADP and a phosphate group. It also will provide the energy for muscle contraction or for chemical reactions. Sometimes energy is needed for chemical reactions. Vice versa, some chemical reactions can release energy that can be stored in ATP. We'll see that as well. All right, so that's all that we have for chapter 2. That's our discussion on chemicals. I hope you've enjoyed, and I'll see you guys soon.